Greetings, students. So, in our last screencast, we talked about how light energy is captured and used to make chemical energy. And so, what happens is the chloroplasts are hit by light, and that light could be from the sun, it could be from a lamp for a house plant. But light hits them, and when light hits the photosystems, then electrons are excited. So, here's an electron being excited from photosystem 2, here's an electron being excited from photosystem 1. And then what happens is the electron in photosystem 2 is passed down an electron transport chain, proton gradient is generated, and that's used to make ATP. The final electron acceptor is photosystem 1 because its electron had previously been excited. Photosystem 1 passes its electron to NADP plus to make NADPH. Meanwhile, we have to replace the initial electron from photosystem 2. That's done by water, and when it does that, it becomes oxygen. So here's just kind of another look at that. Photosystem 2, electron transport chain. Photosystem 1, here's NADP plus being formed into NADPH. And then the ATP being produced. So we've captured the light energy, and we've converted that, so that's the first question, and then we've converted that into chemical energy, specifically ATP and NADPH. So there's one final question we need to address, and that is, how do we use that chemical energy now to make glucose? Well, introducing the light-independent reactions. They're light independent because they do not require light. They can happen in the light, but they can also happen in the dark. So light-independent reactions use the energy of ATP and NADPH to produce carbohydrates. The light-independent reactions are also called the Calvin cycle. They used to be called the dark reactions, but that term has sort of grown out of favor because, again, they can happen in the light. They just don't need the light. So dark reactions, we don't like that term anymore. So these occur in the stroma. And the stroma, you may recall, is the interior of the chloroplasts. And so the process is shown in this diagram, and you can see this is a fairly complex process, similar to the Krebs cycle in a lot of ways. And you actually have a picture of this in that packet that we use for cellular respiration. So we're not going to go through it in quite as much detail. So here's kind of the gist. Basically, carbon comes in as carbon dioxide, and it comes out as this 3-carbon unit that is then used to build sugar and other organic molecules. So how does it work? Well, the first step is called carbon fixation. Carbon fixation means we're sort of taking CO2 from the air and attaching it to a biological molecule. And so what happens is there's this 5-carbon compound. I believe it's ribulose bisphosphate is what that's called. You don't need to know that name. And an enzyme called Rubisco takes carbon dioxide from the air and it adds it to this ribulose bisphosphate to create this compound. Okay, so the act of taking this CO2 from the air and introducing it to a biological compound, that's called carbon fixation. And Rubisco, I believe, fun fact, is the most common enzyme in the entire world, because plants are photosynthesizing all over the place. So that's a big one, Rubisco. What happens then is the carbon dioxide molecules that are brought in are sort of attached, and then a series of chemical reactions take place. Note that ATP is being used and NADPH is being used for these chemical reactions. And ultimately, we get this 3-carbon unit called glyceraldehyde 3-phosphate produced. After that, the starting material is regenerated. And that's why we call it a cycle, because we start with that ribulose bisphosphate and then we regenerate it at the end. So that was quick, but basically just understand this concept that we take carbon dioxide in and we take sugars out. And to do that conversion, we're using the chemical energy of the ATP and NADPH that was produced during the light-dependent reactions. So this accounts for where carbon dioxide is used and where the sugars are produced in this process. So now we understand water was that electron donor. Oxygen is produced after it gives off its electron. CO2 is used in the Calvin cycle. The sugars are produced at the end. And note that it's actually a 3-carbon unit that's the immediate product of the Calvin cycle, but then that's used to make this glucose. So that's pretty much it. At this point, we have successfully used light energy to make sugars, which is pretty incredible when you think about it. Thanks.